In Chapter 3, we're going to take a closer look at the innate immune system. We've learned that the overall immune system basically has two arms, innate immunity and adaptive immunity. The defenses included in the innate immune system are those that act immediately whenever a host is attacked by a pathogen. It's fully active and functional at birth. It's so efficient that most pathogens are destroyed before they even enter the body, or if they do get into the body, they're destroyed before they can multiply and cause infection. We're going to look at two different aspects of the innate immune system. Those that we consider the external defense system. These are primarily anatomical barriers that keep organisms from gaining entry. And then the internal defense portion, which is what goes into effect in the situation where the organism does gain entrance. Our external defenses are largely barrier-based. Some of the barriers have secretions associated with them, which add an extra layer of protection. And then interior defense system includes things called pathogen recognition receptors, acute phase reactants, the inflammatory process, the process of phagocytosis, and some special cells known as natural killer cells. And of course, we're going to look at all this in more detail. The external defense system is composed of physical, chemical, and biological barriers that all work together to prevent infection from entering the body. Now, one of the first and foremost and probably most significant aspect of this is intact skin. The epithelial cells are very closely bound together, form a nice tight layer that very few microorganisms can penetrate. The skin is a phenomenal defense system, starting with the fact that epithelial cells are packed tightly together. There's really no space in between the cells. The cells are filled with keratin, which is this rough, tough protein that makes it fairly difficult to tear or break this barrier. Once we go a little bit deeper into the dermis, which is a connective tissue, there's blood vessels, hair follicles, sebaceous glands, sweat glands, and cells. So lots of weapons and warriors in the dermis, including the macrophages, the dendritic cells, both of these which are phenomenal at presenting antigens to T cells. And also within the dermis, of course, there's secretions and chemicals that are by themselves antimicrobial. They inhibit microbial reproduction, either due to a fairly low or acidic pH, and other mechanisms by which they inhibit the reproduction of the organisms that do gain entrance into the dermis. The respiratory tract is a key player in association with the external defense system. It is lined with mucous membranes, and the mucus produced by those mucous membranes it's physically sticky, and it traps bacteria and other pathogens from gaining entrance deep into the body. And once the potential pathogen is stuck in the mucus gets brought up and out of the body, typically coughing and sneezing, which physically removes them and prevents their access from deep in the respiratory tree. The urine is helpful in protecting us in that we have this flushing action every time we empty the bladder. We flush out any organisms that have gained entry via the urethra. The high acidity of portions of the digestive tract help prevent the growth of many organisms, as well as the mucous membrane, both at the oral end of the system and the anal end. And lastly, the normal flora, also known as the microbiota. We have known for as long as I've been around how important our normal flora is. But in recent years, the research has really taken off and drawn many associations between uh, the appropriate normal flora and various disease states. And that research continues, and I think there's going to be some more earth-shattering data that comes out of those. But for now, we'll just keep it quite general. And one way that I think about this process is a lawn. So if your front lawn is thick and lush and full of grass, it is much less likely to acquire weeds. I mean, they'll sneak in periodically, but by and large, the presence of all this good stuff keeps the bad guys from getting in and establishing a foothold. If you have in your front yard nothing but dirt, okay, there's no luxurious growth of grass, then within a very short period of time, it'll be full of weeds because whatever lands there finds appropriate living conditions and is free to multiply like crazy. So within our body, there are a number of areas of our body that are supposed to have lush lawns, if you will. They should be coated and just full of normal organisms, such that when pathogens happen by, they show up in the area, chances are good that they can't find a place to land, a place to stick to and live and thrive because of the competition with the good organisms. A brief review, kind of putting it all together, all these wonderful structures and mechanisms that we have that are able to prevent the penetration of organisms and kill them in various ways, as well as to physically flush them away via urine, reflexes that are in place, such as coughs and sneezes, that physically expel them from the body. Moving from the external to the internal defense system, we're going to be talking about things such as pathogen recognition receptors, or PRRs, acute phase reactants, the process of inflammation, the process of phagocytosis, as well as the presence of natural killer cells. The PRRs are pathogen recognition receptors. These are receptors that are on host cells that recognize molecules unique to infectious organisms. So in other words, there's something that a given organism possesses in terms of an external molecule that is different from anything that we have. And so this unique molecule is recognized by pathogen recognition receptors. These receptors are found on the macrophages, dendritic cells, neutrophils, eosinophils, monocytes, mast cells, T cells, and epithelial cells. So the vast majority of the cells in our body have these receptors that are able to recognize foreign organisms. Of this list, the macrophages and the dendritic cells are the most important. Sometimes these are referred to as the professional recognition cells. The PRRs are encoded by the host DNA to sense extracellular infection. In other words, these are pathogens that are not inside the host cells, but rather they're in the extracellular environment. The PRRs recognize something we call PAMPs. The PAMP is just the shorthand for the pathogen-associated molecular pattern. So the PAMP, this unique individual or a unique molecule on the pathogen, the PAMP is recognized by the PRR, which is the receptor on the host cell. So when the PAMPs and the PRRs associate with one another, they bind chemically, and this activates the phagocytic cells. Let's look at different types or examples of pathogen recognition receptors. Perhaps the ones that are most well known are referred to as toll-like receptors, TLRs. There's several types of TLRs in humans. Some are on the surface of cells and some are found in the cytoplasm. Then we have receptors known as C-type lectin receptors. These bind to mannan and the beta-glucans found in fungal cell walls to activate cytokine and chemokine production. So in other words, these C-type lectin receptors appear to be specifically in place to recognize fungal invaders. They're the RLR receptor, retinoic acid inducible eye-like receptor. And lastly, nucleotide binding oligomerization domain receptor, or the NOD. Of those receptor types we just listed, the only ones we'll look at any closer really are the toll-like receptors. These toll-like receptors are glycoproteins that bind to substances. So here we're looking at them here, whereby the receptor is very firmly anchored all the way through the phospholipid bilayer that makes up the plasma membrane. These TLRs, when they recognize an invader, they activate cytokine and chemokine production, chemical messengers, and other processes to enhance phagocytosis. This process can destroy most pathogens that we're exposed to before infection sets in. And just to make the distinction, we can have organisms within us that are not causing infection. And one way to define infection, rather than just organisms present, is organisms that are present and multiplying to the point where they're causing disease. There are a group of molecules that we refer to as acute phase reactants. These are soluble molecules that are found normally in serum, but they increase rapidly in response to infection, injury, or tissue trauma. What do? Well, some of them facilitate contact between the microbes and the phagocytic cells. They help those two constituents get together to enhance phagocytosis. Others mop up and recycle important proteins after the phagocytosis has occurred. In other words, if something is reusable, we want to capture it in some way, shape, or form and put it to use again. And of course, if it's not usable, then we want to get rid of it. Here's the table that you see in your book listing the most common acute phase reactants. Now they have them listed and then spend, the time to think about how long it takes for them to respond, the normal concentration, and how high they increase when they're activated. I've made you another chart that you can find on Blackboard that I think is, in my way of thinking, it's more helpful, where I want you to associate primarily the protein with its function, consider whether it's mopping up or whether it's recycling constituents, and then consider how we use this in the lab. What is the clinical significance? Do we measure it? If we do, when might we expect to see an increase in this? So do take a look at that chart. I would strongly recommend that you fill that out, and hopefully that'll help you get some perspective on just what these things do and how they operate when they are called into action. Here we want to think about inflammation for a moment. Um, what I note about inflammation typically is that in the public, they some people tend to conflate inflammation with infection. Infection does involve inflammation, but inflammation does not necessarily indicate infection. It can certainly be caused by other things. And I think in the public, sometimes if you try to probe their minds, they have an association with inflammation as being a bad thing. Because when it's out of hand, when it becomes chronic or when it's very severe, it produces noticeable side effects. But, at this point, I just want to emphasize inflammation is part of the healing and the recovery process. It's a good thing. Okay. It helps us um, get healthy and remain healthy as long as it's activated appropriately and the process kind of runs through to completion at the appropriate time and the inflammatory process shuts off. So you've probably seen this before, but we want to reemphasize the cardinal signs and symptoms of inflammation. Some references will refer to 4 cardinal signs. There's a 5th one in there that doesn't always apply. Kind of depends on what part of the body the inflammation is occurring in. So the little graphic I think is cute to kind of remind us what the cardinal signs and symptoms of inflammation are. Heat, redness, also known as erythema, red skin, swelling, which is edema, pain. Those are the 4 big ones that everybody has on their list of inflammation. And then the last one is the loss of function. And this applies primarily to any extremities. And just from the standpoint that if you have inflammation going on in a finger or a hand or an arm or a leg, etc., you are not going to want to use it. Why? Because it hurts. So you naturally take it out of service to the degree possible and wait and hope that the pain and the redness and the swelling goes away. Here we look a little more closely at just the very beginning stage of inflammation, where we've got something going on out here in the tissue. Over here, this is in the bloodstream. Okay, so something happens, and the alarm is fired. And so what happens ultimately is due to the release of various chemical messengers referred to as selectins and chemokines and integrins, etc. I don't want to get too hung up in too much detail with this. But the final result is that macrophages and mast cells get the signal that they're going to leave the circulating blood. And the way they leave the circulating blood is to squeeze in between 2 adjacent cells. And this process of squeezing through cells, going from the blood through the vessel wall into the tissue, is called diapedesis. Diapedesis, the word literally means to walk through or walk across. And so once this process gets started, then more and more white blood cells are brought to the area and begin the process of phagocytosis and other inflammatory processes that result, hopefully, in the destruction of the pathogen before an infection begins. Here we look at the process of phagocytosis using the graphic from your textbook. And so this is our generic phagocytic cell. Now, the word phagocytosis literally translates to a condition of a cell that eats. And so this eating cell, this phagocyte, has made contact with a bacterium. Upon contact, the membrane of the phagocytic cell starts to invaginate or kind of pouch in, and simultaneously, like kind of like arms of this membrane reach out as well. And when those arms reach one another, they fuse. And ultimately, what we have is now inside the phagocyte, we have a membrane bound bacterium, and we call that the phagosome. Also, within the cytoplasm of this cell are membrane-bound organelles called lysosomes, and lysosomes contain digestive enzymes. Now, in 4, we see where a lysosome has made contact with the phagosome and has opened up, allowing the contents of the lysosome to enter the phagosome. And in 5, we have a phagolysosome. So now we have this bacteria kind of trapped in this pouch with these digestive enzymes, and those enzymes start breaking down all the components of the pathogen. When that process is complete, when the pathogen is digested chemically, then the waste products are brought to the inside of the host cell membrane, and the waste is released via the process of exocytosis. Now, I've posted on Blackboard what I consider to be an excellent video showing the steps of phagocytosis. And if you'll note on your weekly objectives this week, you are expected to know the steps and be able to describe what occurs in each step of phagocytosis. So you really do need to go to that video and spend 8 minutes, and you'll get a lot of review material of things that we've covered so far in this PowerPoint. Now we look at our NK cells, which stands for natural killer cells. I like to use a little graphic of kind of this little cell as a little Rambo, fully locked and loaded, lots of ammunition. He is a lean, mean fighting machine, and he is ever at the ready to get rid of the bad guys for us. These Rambo cells, these natural killer cells, are very effective when it comes to viral eradication. And they also interact and help us get rid of intracellular pathogens. Now, these two are related from the standpoint that viruses are all intracellular pathogens. Viruses can't replicate or reproduce without being inside a living host cell. There are certain bacteria and other organisms, such as some parasites, that are also intracellular pathogens. In other words, they need to be inside a host cell in order to replicate. So these natural killer cells are great at dealing with all of the intracellular pathogens, viruses, and the others, as well as cells that are self cells, but they've become cancerous. They're tumor cells. And in a nutshell, a tumor cell is a cell that won't stop reproducing or replicating. Normally, a cell divides and multiplies appropriately. But when a cell mutates and it becomes cancerous, it ignores the stop signs and it just keeps multiplying and multiplying and multiplying. Well, the NK cells also can recognize these tumor cells, and it is thought that we all develop tumor cells perhaps multiple times a day, but that because of our active, effective immune system, we almost always get rid of them before they cause a problem. It's the rare exception when they aren't gotten rid of at an early state, which then allows the development of cancer. So what do these NK cells do? They recognize any damaged cell and they can get rid of it. This does not require previous exposure. They just zap it when they see it. What gets them revved up? Well, the release of cytokines that may have been given off by phagocytes that have also been trying to get rid of these organisms, and other mechanisms that stimulate other cells of the immune system to produce these chemical messages, cytokines, that tell the NK cells to get up and get busy and get active. When they get the message to get busy, they also start producing cytokines and other factors that further enhance and enlarge the immune response. So they're involved at the beginning stages of this thing. You know, they're kind of, in some cases, the first responders, but they're also sending out signals to the rest of the immune system saying, hey, we got a problem over here. We need more weapons. We need more fighters over here to take care of this. These guys are involved in both the innate immune system. How are they involved in the innate immune system? Well, they're there. They're ready to go. They don't require previous exposure. They get the signal and they start killing abnormal cells. They're also involved in adaptive immunity in that as they are doing their job, they're sending signals that ultimately lead to the production of lymphocytes and sensitized T cells and B cells. So the NK cells are constantly surveying host cells. They approach a host cell if there's MHC class 1 protein on the outside of the host cell, but there's no foreign protein or other stress proteins associated with that MHC1 protein, then inhibitory signal is sent to the NK cell telling it, back off, you know, everything's fine here, nothing to see here, no killing occurs, and the normal cell is released. On the other hand, if an activating receptor is engaged by a foreign protein or a stress protein and class 1 MHC is missing, no inhibitory signal is given, molecules called granzymes and perforins are released and the infected or diseased cell is eliminated by apoptosis. Now, one other situation could occur, and that's when we have a target cell, an infected cell, that is coated with antibodies. And in that case, what results is referred to as antibody-dependent cell cytotoxicity. NK cells participate in this. When an NK cell finds a target cell that has antibody already attached to it, then the NK cell has certain receptors called CD16 receptors that bind that antibody that's already hooked to the targeted cell and again causes the release of perforins and granzymes. Now, the NK cells aren't the only ones that participate in this antibody-dependent cell cytotoxicity. Any target cell that's coated with antibody falls into this category, and besides the NK cells, monocytes, macrophages, and neutrophils can also participate in this. In other words, all these cell types, when they encounter an antibody-coated target cell, they can attach via the appropriate receptor. This leads to the release of the perforins and granzymes, and that leads to the destruction of the infected cell. To review, the innate immune system involves the body's normally present defense mechanisms. Innate immunity is characterized by lack of specificity. There's no need for a prior exposure, And basically a similar response is generated with each exposure. There are external defenses, which are structural barriers and their associated secretions that keep microorganisms from entering the body. The internal defenses include phagocytic cells and acute phase reactants. Pathogen recognition receptors are molecules on the host cells that recognize substances that are found only on pathogens. Once the receptors bind a pathogen, phagocytosis can take place. Pathogen-associated molecular patterns are those molecules on the pathogens that the PRRs recognize. This allows the host to distinguish self from non-self. The acute phase reactants are serum proteins that increase rapidly in response to infection or injury. And inflammation is the body's response to injury or invasion by a pathogen. Natural killer cells kill target cells that are infected with a virus or other intracellular pathogen or tumor cells without having been previously exposed to them. The NK cells are a link between the innate and the adaptive immune response. And so here we see a nice graphic showing you, on one side, the innate immune components, which we've discussed. We haven't talked about the adaptive side yet, but in the middle here, the most important one I want to point out to you folks is the NK cells, which are part of both the innate and the adaptive immune system. They're part of the linkage between the two systems. And now it's up to you to hit the books. And I do want to emphasize, as always, do use your textbook. This is a well-written textbook, in my opinion. I feel it's readily understandable. I've tried to hit the high points of things with you here in this presentation, but don't neglect your textbook as well. Allow it to fill in some of the gaps, and obviously there's more pictures and illustrations and things of that nature in there too that will help you learn and process the material.